Well, so let's begin. I am pleased to welcome everyone for our online school on applied population genetics and analysis. So today it will be an introductory lecture. So our three lecturers are Rasmus Nielsen, professor in UC Berkeley and the University of Copenhagen. Russell Corbett-Detig, professor in the UC Santa Cruz, and Professor Thorfinn Korneliussen in the University of Copenhagen. So thank you very much for our lecturers to agree to teach us. And let's begin with Professor Nielsen.
Okay, let me share my screen. I don't seem to be able to get the option for screen sharing. Have you out screen sharing?
Let me double check it.
Oh, maybe oh, here it is. Sorry.
Yeah.
It's my fault. Okay. All right. So as Vladimir just mentioned, my name is Rasmus Nielsen, and I'm a professor at University of California in Berkeley, where I'm sitting right now in the smoke from the fires here in Berkeley, here in California. And I'm also a professor at the University of Copenhagen, where I spend the summers. And I study the math and the applications and analysis of population genetic data. And so what this course is about is basically about this topic. And there will be these three segments of the class. I'll first teach a segment on what population genetics really is about, and also some of the theory in population genetics, mostly concentrating on getting some of the theory in place so that then Ross and Thorfinn can talk about more practical handling of data afterwards. They will talk about methods for analyzing population genetic data and how particularly to do much of the bioinformatics. The course will be very focused on computational methods on bioinformatics, but the first third of the course I'll be teaching, and I'll be talking more about more general issues, and particularly about population genetic theory. And I thought today I would just give you a little bit more of an introduction to the topic of population genetics. So you know, what is this class about? What are the kind of things people are doing in population genetics? When we use the word population genetics, what do we mean? So the population genetics, what is it? You could define it as the study of genetic variation within and between populations. So instead of, for example, studying the genetics of just one individual, you're studying the genetics of multiple individuals and compare them. And what can we then learn from that? That's what population genetics is about. Population genetics is really the core theory of two different research areas. And one is medical genetics, where population genetics is used very heavily there, because to understand disease causes, what's called disease etiology, and understand the genetics of that, you need to compare different individuals. So that's inherently population genetics. So what people do in medical genetics is, in a very, very simplest case, they take two groups of people, the cases and the controls. And then they sequence some DNA in these two groups of people. So in a particular part of their genome, some individuals might have a C, all individuals have an A. And then they compare the cases and controls and look at, is there a difference in the DNA? In this case, it's the frequency of the A version of the gene, of the DNA, what's called the allele, the A allele. Is that different in the cases, frequency of that, than in the controls? If there's a difference in the frequency of this particular genetic variant in the cases and controls, it's associated with the disease. And it may possibly be because it is part of causing the disease, or something nearby it, or the genus in is causing the disease. So that trick, what's called association studies or association mapping, is used time and time again in human genetics. It's in some sense the core engine of human genetics. And much of the theory and methods behind that, that's population genetics. So what you'll see in many, many publications in medical journals are plots like that. This is called a Manhattan plot. And what you have out of the x-axis here are the different chromosomes in the human genome. So remember, the DNA in the human genome is organized into 22 chromosomes plus an X and a Y chromosome, where the genes that determine your sex, whether you're male or female, they sit on the X and the Y chromosome. So on the x-axis here, you have the different chromosome. You have the first chromosome, the second chromosome, and the third chromosome. Then you do a test to see, is a genetic variant that's in a particular position, is that associated with the disease? So it's in a statistical test, you test for a correlation between the genetic variant and whether or not people have the disease. And then you plot here minus the log 10 p-value. So the higher the point lies on this, the more evidence there is that there's an association between the genetic marker and the disease. And so now you can go along the entire genome and you can figure out, where in the genome do we find genetic variants associated with a disease? And see here, there's a big peak here. So in this particular study, there's a genetic variant associated with a disease there. So that's how you find diseases associated with type 2 diabetes, cardiovascular disease, psychiatric diseases, and so on. And there are large projects all over the world on doing this. One of the biggest projects, so because of these large projects, you now have these big catalogs of where in the human genome to find genetic variants associated with diseases. So this is an old plot from 2012, just looking at 18 traits. Now you have thousands of traits and hundreds of thousands of positions that may or may not matter for a disease. And you can see for each particular position in the chromosome, you can look at, is there a genetic variant there associated with a particular disease? So we have big catalogs from that. The biggest data set that people are working on right now for this kind of thing is the UK Biobank. So this is an initiative funded by the Wellcome Trust, where they have initially 500,000 people, where for these 500,000 people, they have the genomic information, they have sequenced their genomes, and they have all of these, what we call phenotypes. That's something that's measurable from the person. So that's physical measures, how tall are you? It's all kind of biological, physiological measures. They measure physical activity. They do all kind of imaging on people. They do all kind of cognitive functions. They have also lots of self-reported data where people report for themselves, all kind of things about their behavior. So because of this is all public data. Now you can go in human genetics, you can go in and ask, if I'm interested in particular traits, what determines genetically that trait? So for example, the databases for this, so we can go in and we can ask, who likes to drink beer? What determines whether you like to drink beer, for example? And then you can go see a plot, again, a Manhattan plot for beer, and find, there are a couple of positions in the genome where the genetic variants associated with beer drinking. People that have a certain genetic variant in that particular position of the genome, they're more likely to drink beer. So, and then you can ask, what are the genes there? So then you go down, there's a list here. It's a little bit, the font is small, so you maybe can't see it, but the first gene that appears is a gene that's called alcohol dehydrogenase 1B. So that's a gene that determines how well you can metabolize the alcohol, the ethanol. If you have a certain version of that, then instead of metabolizing it easily, you will get what's by some people called the Asian alcohol flush syndrome. You will sort of get all flushed, and it's a little unpleasant to drink alcohol because you essentially get the hangover before you get drunk. That's no fun to get that, just the hangover. So people that have that genetic variant, they don't tend to drink so much beer. If you go down this next one, that's one that's called FTO. That's another gene. That gene is associated, generally, very important gene that's associated with obesity, with being fat. And that's because it controls appetite. It helps control appetite. So beer drinkers, what genetically determine how much beer you're drinking is whether you have this sort of intolerance due to this ADH locus, and whether in general you're glutton, whether you like to drink, and eat a lot. If you do that, you tend to drink more beer. That's what this study tells us anyway. So you can then ask about other things. For example, you can ask now, what about other forms of alcohol? What about red wine? Now for red wine, the story is somewhat similar in that this thing, people that are have this alcohol intolerance. They also don't like to drink a lot of wine. So if you don't metabolize alcohol well, ethanol well, you don't drink a lot of red wine. But what's the second locus? It's not this FTO locus that has to do with gluttony, with just eating a lot. No, it's a taste receptor. So if you have a fine sense of taste, you're more likely to be a red wine drinker. Okay, so you can believe that story or not, but that is essentially the kind of information. It's just sort of a bit silly example of the kind of information that people are retrieving now from these data. Of course, we're really most interested in diseases, finding the genetic variants associated with that, so we understand why diseases are happening, the disease etiology, but we also get information about all kind of other things in humans. So that's what's called association mapping. It's used in medical genetics, and population genetics in some sense forms a theoretical foundation under that, on understanding how is human genetic variation distributed and what causes certain patterns of human genetic variation. And we use that to understand power in these, statistical power in these studies, correlations between different genetic markers and other things like that. So understanding population genetics is important for these kind of studies and forms the basis, the theoretical basis for that. But population genetics is perhaps more important or even more central in the study of evolution. In some sense, population genetics forms the theoretical basis for the study of evolution. It tells us how populations are evolving. And that's, in some sense, is the basis for all understanding of evolution. So it also is a theoretical foundation for evolutionary biology. And it is used extensively to understand evolution and also things like human history. So, for example, we had a paper come out yesterday on how Vikings, they distributed all over the world, into Russia, for example. Swedish Vikings, they sailed down the Volga and they affected, the genetic variation in Russia today. And many places all over the world, they certainly affected very strongly the genetic variation in England that was occupied and colonized by the Vikings. That's back in the days where the people in the United Kingdom, they were the ones being colonized by other people. It's quite a long time ago that happened. And you can see today that the Vikings have made a genetic imprint there. So you also use it for those kind of studies to understand human history. Okay, so from these studies, we learn a lot of different things about how people have moved around in the world. And we'll be talking a lot of how can you, from having some DNA sequences, make inferences about this kind of stuff? How can you know how people moved around the world from today looking at some DNA sequences? There's a lot of modeling and statistics and computation going into that we'll be talking about. The basic models are based on modeling genetic variation within a population. So we'll talk a lot about models that look like this. The oval here, that's a population. Within that population, we have some individuals. Those are the little circles in here. And then we color them, for example, 2 different colors, white and black. That's because they differ in a position in the genome in some genetic marker. Okay, so there's a difference between them. And we're interested in through time. So now we're looking at a different time point, a specific time point here. But we're interested in how does this evolve through time? How would the frequency of white and black, how would that change through time? And models of that, stochastic process models of that, forms the basis for the kind of predictions that people are doing in evolutionary biology to infer how people move around the world, how the Vikings, they settled the United Kingdom and also moved down the Volga River and so on. So the process by which these changes happen is called genetic drift. So genetic drift is a term that describes the stochastic changes in the frequency of a genetic variant between generations. So if currently a genetic variant is a frequency of 40% in a population, in the next generation, because of random events of who has offspring and how you transmit your genes from parent to offspring, it might not be exactly 40% in the next generation. There might be a little fluctuation. So we can describe that if we look through time for the number of generations and look at the frequency of a particular genetic variant, we can describe the path by which it changes through time. And we can develop mathematical models for that, for how this genetic drift, the change in the frequency of genetic variants, how that happens through time. And again, that's in some sense, that kind of model is the basis of the foundation of what we do in population genetics. That's how we understand genetic variation. Genetic variation comes from first a mutation arises that introduce new variation, and then this genetic drift can affect the frequency of it. It can increase in frequency. So for example, you have here the green variant here that is increased in frequency in a period here. So that's the reason why genetically, we're not all the same. We look different from each other. Why there are hundreds of thousands or millions of positions in our genomes where we're different from each other because of this process, predominantly because of this process of mutation and genetic drift. So we'll be talking a lot about mutation and genetic drift in the first couple of classes of this course to understand this process better. What's going to lead it to is a body of theory that was developed in the 80s and the 90s that calls coalescence theory. And the idea there is that from these kind of processes, following them forward in time, you can construct a process backwards in time, a dual of the forward process, by which you follow the history of a sample backwards in time. And that forms the correlation amongst individuals, or the relatedness among individuals, can be described by a tree-like structure that's called a coalescence tree. So we'll be discussing that. Where does that coalescence tree come from, and what can it be used to? It's basically the structure that we use to describe relationships between individuals and also genetic similarities between individuals. We can describe that through these coalescence trees. So we're going to talk a lot about where these coalescence trees come from and what we use them for as part of this as well. And then we're going to go back to look at this question of how have people spread around the world and relate these trees to that question, and show this through inference of these trees, essentially, that we learn a lot about this particular question. Okay, so now I describe everything as just being a matter of these mutation and then these random fluctuations that causes changes in the frequency of genetic variants through time. But there's also other important processes in population genetics that affects this, in particular, natural selection. Okay, so what is natural selection? I believe you're probably all familiar with the idea about selection from Charles Darwin. You know, from the origin of species, he argued that similarly to what's going on in domesticated species, where humans have selected certain breeds of horses and certain breeds of dogs by every generation, choosing the dog or the horse that they most prefer and allowing that one to breed. Something similar is going on in nature, where some individuals that have lower fitness, they will not leave as many offspring in the next generation as other individuals that have higher fitness because of certain traits that they have. And because of that, these traits will change through time. Okay, so that's what we call natural selection. And it's an additional process in addition to genetic drift that also affects these changes in frequencies of genetic variants through time. So we're also going to be able to try to talk a lot about that. How does natural selection affect these processes? And why are we interested in that? Well, because then, when first we understand that, we can go back to the data and ask the question, how do we actually identify where there's been natural selection in a genome, in a certain species, for example, humans. So we can go back and ask, when humans, has natural selection changed our genetic makeup within the last, say, 5 or 10,000 years? What are those positions? So similar to the Manhattan plot I showed before for finding associations between a disease and genetic variants, we can make similar plots along the length of the genome, where we find positions in the genome where there's an association between genetic variants and selection, or rather where there's been natural selection acting on genetic variants in the genome. So we can do what's called a selective scan, where we identify regions in the genome under selection, present or past. And we're interested in that because selection really tells us something about function as well. And the reason why it does that is, if a lot of people are producing fewer offspring, perhaps because they don't live to become adult, or because they have certain genetic variants, those genetic variants are probably very, very important for something associated with diseases, otherwise very strongly affecting how humans function. So if we can look at and identify whether it's natural selection, then we also will learn something about function from that. That's one reason. The other reason is we are interested in how did human, how did we come to be as we are today? So we're interested in our history, and it's also important for that. Okay. So in this plot, as you can see, a little valley here, since it shows a little and no evidence for selection, that will change the function. And then if you have a peak like that, that shows strong evidence for natural selection. Okay, well, maybe I'll go through this example here. So this is a peak here. There's a really, really strong peak, a particular place in the genome, if you look at Europeans. And the strongest peak that you tend to get in most studies is for a particular gene or locus called the LCT, which is a lactase locus. So this particular gene encodes a protein that is called lactase. What does lactase do? Well, it is the enzyme that allows us to digest lactose, the carbohydrate that is in milk. So the reason why babies can drink milk and get all the energy out of that is because they produce a lot of this protein produced by this gene. And the reason why some people, when they're adults, they become lactose intolerant, they can't drink milk, is because they stop producing a lot of the product of this gene. They stop expressing that. It's no longer active, that gene. Okay. And so they don't produce lactase, and therefore they can't digest milk. Now there's difference between people. Most people in Northern Europe, particularly around Scandinavia, but also other places, including Russia, they're not lactose intolerant, most people. They have what's called lactase persistence. There's a particular genetic variant here by which they have, because they have that variant, they continue to produce lactase. The gene continues to be active when they're adults, and therefore they can keep drinking milk. But if you go, for example, to Southeast Asia, almost nobody have that genetic variant. And because of that, they can't produce lactase as adults. Most people there have some form of lactose intolerance. And what we think is going on is that there's been natural selection in Europe for increasing the frequency of the genetic variant that causes lactase persistence, avoidance of intolerance. There's been natural selection for that after the introduction of dairy farming, after you started having cows and drinking milk. The ones that actually could drink the milk and not get sick from that, they had an advantage, especially sort of in a cold Russian or Scandinavian winter. It would be a great advantage that you could continue to get some proteins by drinking milk. Okay, and people that couldn't really do that, that became sick from that, they would have a fitness disadvantage. And we see that in the genome. We can find the signals of that in the genome. We can see that in Northern Europe, there's been very strong natural selection over the past 3,000 years in this genetic region where the lactase gene is sitting. Okay, so that was the story about the lactose gene. But there are many other stories. For example, now we're thinking about a lot of these days about viruses. We have the COVID-19 virus and so on. And you might be interested in what's going on there. Well, one of the reasons why viruses are so hard for us medically to deal with is that they evolve very fast. So, for example, the influenza, even though you get vaccinated every year, you can still keep getting influenza. It's not enough to get a vaccine once. Why is that? That's because influenza evolves really fast. So you might be immune to one version of influenza, but then it evolves and you might be immune to another version. And one of the things we're worried about with COVID-19, of course, is does it evolve fast enough that we'll have that problem for COVID-19 as well? Its mutation rate is about 10 times lower than that of influenza. So maybe it doesn't evolve fast enough. We hope. You can just get vaccinated once if we find a vaccine that's effective enough. I know there's a lot you discuss in Russia right now about the vaccines and so on. So in any case, so you can then ask what's going on in the virus. And so this is for the influenza. So this what you have down here to the right is the influenza particle. And the little sort of pegs that come out of the particle, these are molecules that are called hemoglobin molecules. They sit on the outside of the virus particle. And what you can do is you can look at the DNA sequences of the virus and figure out what are the positions that evolve very fast. And to the left, there's a cartoon of this protein on the outside. And the parts of it that's in red, that's where it's been evolving very, very fast to escape the immune system. Okay, so to escape the effect of getting a vaccine. And what we can read that off the DNA sequences in the virus. We can use population genetic methods to infer where has there been natural selection. And by that, we can find the residues that have been interacting with the immune system and that are part of this evasion of the immune system. And that's something medical researchers are using all the time to when they design new vaccines, for example, to try to say, these residues that evolve all the time, maybe we shouldn't target those. We should target some other residues. That's another example of how these kind of analysis are being used. I'm going to give a couple more analysis of this about natural selection and sort of motivation for why we're doing all this. Tell some stories about how all this research is being done. So one thing from our own group that we've been doing a lot, we worked on altitude adaptation in Tibet. how can Tibetans function so well in the high altitude plateau in Tibet at an altitude of more than 4 kilometers height, where the pressure, oxygen pressure, is only 60% of what it is at sea level. Tibetans, they function really well there, and other people, they have a tendency to get chronic high altitude sickness. Why is that? Why is that this difference between the Tibetans and other people? Well, what we did there, we compared people that are Han Chinese and people that are Tibetans, and then we looked at the frequency of different genetic variants. And the way to read this plot is that you have different genetic variants here, and if you are far out on the x-axis, the frequency of that variant is high in Tibet, and if you go up the y-axis, the frequency is high in the Han Chinese, the most common ethnic group in China, as a reference population. And what we find was that there's some particular genetic variants that have very high frequency in the Tibetans, but relatively low frequency in the Han Chinese. And those ones we could then later show, those are actually the ones that changes some of the physiology of the Tibetans. So they produce red blood cells differently, so that they become much less likely to get a high altitude sickness. In that way, you can find these genetic variants by looking at where there's this is the sign of natural selection in the Tibetans. The frequency of these variants have increased in Tibetans, and we can then, by identifying what's under natural selection, we can then find genetic variants that are very important for specific function, in this case, high altitude sickness and other forms of performance in lower oxygen environments. All right, I'll give one more example, a last example of this, what we can do with natural selection. Another example from my own research. I'm going to sneak that in here. This is a project I was part of with some other research groups looking at some people that live in Indonesia, the Bajau sea nomads, which really have a very, very special and unique lifestyle. So we're very interested in, well, do they have any biological adaptations to that lifestyle? Just going to mention a little bit about them. So these Bajau people, they live an entirely marine-dependent existence. They live on boats or coral reefs, and they're traditionally nomadic, and they get all of their food, almost all their food, from diving. They will dive down, they'll find invertebrates, mussels, and so on, and they will also hunt fish, and the majority of their food will be from this diving. Okay, don't know quite how long time they had that lifestyle, but they've existed as a cultural group with that lifestyle. We know from written evidence for at least 1,000 years, because there's a poem that's 1,000 years old where they mentioned, like a Bajau princess was married into some family. So we know they existed for at least 1,000 years, but perhaps much longer than that. They can dive really far down. There's some debate about this. There's anecdotal evidence that the free dive down to 70 meters. But perhaps more important than that, and more relevant, is that anthropologists have been observing them. They've noticed that during their daily working hours, they spend 60% of their time underwater. So imagine that you're out for 8 hours and 60% of the time you're diving. That's just incredible physiological pressure you put on your body. Okay, you're holding your breath most of the time, okay, for 8 hours. So that fundamentally changes a lot of different physiology when you live like that. So is there any biological adaptations to that? Has the biology somehow changed in response to that? Well, you can look at different things. You can, for example, look at the diving reflex. So you may not be familiar with the diving reflex, but everybody has it. All mammals have it, including humans. Or we think all mammals have it at least. And certainly humans have this diving reflex. It's a very evolutionary old reflex that exists. It is induced by apnea and cold water facial immersion. So that means if you put cold water on your face and you hold your breath, then this will happen, this diving reflex. And what does the diving reflex do? It's then transmitted from your face to the rest of your body, and it causes bradycardia. So that is a lowering of the heart rate, which lowers oxygen consumption, so you can hold your breath for a longer time. It causes peripheral vasoconstriction. So that means that the blood is sent into the interior part of your body, to your brain and your interior organs, where it's needed the most. So you won't die so easily if you have low oxygen. And then there's a contraction of the spleen. The spleen is an organ that you have in your body that is a reservoir of both red and white blood cells. But also it contains particularly the red blood cells, which are the ones that transport oxygen in the blood. So when the spleen contracts, red blood cells goes out in your bloodstream, and you can hold your breath for longer time because you now have more oxygenated red blood cells in your bloodstream. So it's like you're having a diving tank with you, essentially, in the spleen, a little internal anatomical diving tank that you can contract whenever you're diving. So these things all go on by when you hold your breath and put cold water in your face. And in fact, you might all be familiar with that feeling. You jump a warm summer day in Russia, you drop into a cold lake, you get cold water in the face, and you feel really refreshed. So that's actually not just sort of something psychologically in your head. That's also this diving reflex that will help induce that feeling because of these specific physiological reactions. So here's a graduate student, Melissa Lado. She went down and measured spleen sizes among people, the Bajau people in Indonesia, and also took genetic samples. And when she measured spleen samples, she could show that the Bajau people on average have larger spleens than other people. And she compared to some other groups in the area that are not diving. Now, you say, oh, so that, okay, maybe shows that the spleen in the Bajau has adapted to become larger because they're diving. It's very important that it does not necessarily mean that. It could be caused by other factors. It could be a plastic response. It could be because they dive more that induce the spleen to become larger. It could be because they are diving places where there are more pathogens. You get enlarged spleens. Certain pathogens also cause the spleen to become enlarged. But it possibly could be a genetic effect. It's a possibility. And so what we did was we scanned their genomes to find variants. Sorry, oops. Now it's all running. We scanned the genomes to find genetic variants that show strong evidence of selection, particularly in the Bajau, and also are associated in these kind of association study with the spleen. So we did both. We looked at something that both have natural selection acting on it, and among those candidates has association with the phenotype. So we do both the things I talked about first, the medical part where you look for association, and the other thing where you look for natural selection, and combine the two. And that led us to one gene that's called PDE10A, which in fact we can, which has a very strong selection signal and also moderate association signal, and that we can show in other studies, in mouse studies, is associated with spleen size. So now we can show that when you, or Melissa has shown that when in a mouse study, when you modulate the expression of this gene, the function of this gene, you modulate also the spleen size in mice. We can also show in other cohorts this gene is associated with the size of the spleen. So what we think is going on in these Bajau people is that as they adapted to this life of diving, of holding their breath all the time, natural selection has favored them having a larger spleen so they can hold more red blood cells, so they could dive better. It's like they have evolved a larger diving tank as they're diving more for this. And we can find the genetic elements that are responsible for that. Okay, so I should mention, so that was natural selection, but I should mention we use population genetics in many other areas. For example, analysis of ancient DNA, that's very relevant now in Russia, where because of global warming, much of Siberia is undergoing a lot of melting. That means that you're recovering a lot of animals from the Pleistocene from which you can get DNA out, and you can then analyze these samples and, for example, learn things like why did certain animals go extinct and why did other animals not go extinct. What you can also do is you can drink samples down through the soil in the permafrost, and people are doing that right now in Siberia, and large grids drilling down, getting samples of soil, and from that extracting DNA. And from that, you can actually, from that DNA, you can look at the populations through time as they have evolved. It's like a time slice in the permafrost, frozen down in the soil of the DNA that's left in the soil that you can use to find out when do populations go extinct, what were the population samples, and many other things. So as part of a study many years ago, I think that did the first time did something like this. It was not for a soil sample, but it was for an ice core in Greenland, where people have been drilling down a long core of ice through the ice sheet of Greenland, in the middle of Greenland. And that the depth of that is almost 500,000 years old. And in that ice is embedded little small particles that have DNA in them. And from that, we can then reconstruct the environment. So we can go and make tables of what plants, for example, were at particular times in this sample. And that way we can reconstruct the environment through time in Greenland. So population genetics is also doing those kind of studies, for example. So this population genetics is used for many kind of things. It's used in medical genetics. It's used in these kind of studies. It's used in many studies of evolution and in several other areas. So end up talking a little, now I talk a lot about biology. In this course, this course really doesn't otherwise talk much about biology, but this sort of gives the biological motivation. But I want to talk a little bit what I think are the prerequisites for this in terms of biology. Another prerequisite is you should know basic probability theory and understand how a computer works and things like that. I'll assume that you know that. But there are also some biology that you need to understand. And so if you're very confused about some of the things I said today, it might be good to go back and look at sort of a high school level textbook on genetics. So we will assume generally that you have high school level genetics. So you should understand what DNA is, for example. What is DNA? When we talk about DNA, what is that? We say a gene. You should understand what we mean by a gene. You should understand what a chromosome is, what cell division is, what a mutation is. When we say a mutation, you should understand what that is. And how DNA is transmitted from parent to offspring is also important. So Mendel's laws. So these are all things that are part of I would say in most countries, I believe also in Russia, part of a high school curriculum in biology to understand these things. And we will assume that you understand this. I'm just going to give you a short little reminder because even for biology students, I find sometimes that's useful. So you should understand what a cell is, that inside the cell there's a nucleus. Inside that, there are chromosomes, and the DNA sequence is organized up organized into these chromosomes. It's wind up to become a chromosome. The DNA sequence itself is this double helix where you have 4 different bases: adenine, A, T, G, and C. G pairs with C, A pairs with T. And the genetic code, when we're talking about that, that's linear sequence of an ordering of A, T, G, and C. Okay, the human genome has a total for all 22 chromosomes plus the 6 chromosomes. It has about 3 billion base pairs like this. So a base pair is, for example, an A that is paired up here with a T, or a G that's paired up here with a C. We call them base pairs because of the double helix structure. They're always in pairs. A has to bind to a T and a G has to bind to a C, and so on. So there are 3 billion of these base pairs in the genome, and that's the genetic code. So you should understand that. To understand what a mutation is, you should understand that you have a DNA sequence here. That's a DNA sequence, like I talked about before, a short little stretch of it. Then when the cell divides and you produce what's called gametes, those are the sex cells, like the egg cell in women and the sperm cell in men. And error can happen in this process. And for example, here, the copying of the DNA from one cell as it divides to the next cell is not perfect. So occasionally you have errors. They can be of many different forms, but the most common form is just that you take a base, a G here, for example, that becomes a C. Okay, so that's an error in replication. That's what a mutation is. So you should understand that concept. You should understand that when you look at DNA sequences from many species, for example, the reasons why there are differences, you see differences between them, you see some differences in the DNA, is ultimately because of this process. They've evolved from a common ancestor, but then errors have happened, and then other things have happened as well, which we'll be talking about, and that causes these differences between species in their DNA, and ultimately explain why a horse is different from a zebra, and it's different from a rat, and it's different from a human. And then you should understand the central dogma of biology, the way that information is being processed. You have the information stored in the DNA. The DNA is being transcribed into RNA, which is inside the cell nucleus, is then being transported outside the cell nucleus to the cytoplasm, where it's being translated into a protein. Okay, so the proteins are the ones that are doing the function, whereas the DNA, mostly the RNA is also doing a little bit of it. We know now, didn't think that 50 years ago, but we know that now. But mostly the function is done by the protein, whereas the DNA stores information. So that whole idea about that sort of basic high school idea about how molecular biology works at the high school level, you should be familiar with that. And if you're not, it's something that you can go back to a high school book or look up on Wikipedia with, look for these topics and study up yourself. It's not that hard. Okay, that's all I had for now. Are there any questions to any of this from anybody?
Well,
no,
like
I think we should then go on to Ross then.
But Rasmus, what is a gene?
We'll be talking more about that later. That's a good question.
Thank you very much, Rasmus. Yeah. So, Russ?
Sure, I can do some stuff. I'll say I had planned this mostly for me. So my goal here is to figure out what you guys know about and also what you want to know about. So those of you that have webcams, I'd like you to turn them on, and we're going to do an activity as we go here. Rasmus and Thorfinn, you can participate as well. But what I'm hoping you'll do is kind of hold up 3 fingers. One is, I have no idea what you're talking about. I've never heard of this before. 2 would be, I'm familiar with this, at least the key words. And a 3 would be something like, this is boring. I don't need to learn any of this. Don't ever teach me this. Does that basically make sense to everybody here? Okay, those of you without a webcam, could you just maybe type that into the chat as we go? Give me a real-time updates. Give me a 1, 2, or 3 about what's going on, just so I have an idea. And the reason we're doing this is that, in a month from now, when Rasmus is done telling you about exciting biology, is when I'll start teaching. And it's going to be the kind of the nuts and bolts of how you get to that exciting biology using the kinds of data that we make today, and a sort of general explanation of how that works. So I need some idea what you want to know, and that would help a lot. Okay, and with that, I will share a screen here. I'm assuming everybody is seeing this screen more or less. Give me a 1, 2, or 3 here. You're actually seeing it. You're kind of seeing it. No? Okay. What's going on there? So I want to do that. Okay. Everybody got a thing? Yep. All right. So as I said, this is going to be the kind of nuts and bolts of how we do population genomics today. And Thorfinn will actually do the back half of that, which is how you go from these kinds of process data objects to actual biological results. I'm mostly going to talk about how you make sense of the raw data itself, where you can get some of that data, how you can use this information to do the kinds of biology you're excited about. Okay. So the basic outline is going to be something like this. It's going to start with just a lecture about genomes, including kind of the biology and variation among them. Not in great detail, but just some of the key issues you need to be aware of that actually inform how we process the data and what makes sense to us when we do that. And we'll talk about that as we go. We'll also talk about the raw data itself. So both how it's generated and then what the differences among data types are, how you can learn from that sort of thing. Okay, and then common sequence repositories. So things like the Sequence Read Archive, but also sort of customized things like the 1000 Genomes Project maintains its own repository of lots and lots of process data, how you can get to those kinds of things, and how you can use them to make inferences. Okay, we'll also talk about data generation and processing. So again, the sequencing technologies themselves, how they work. Processing the raw data. So unfortunately, what streams off your sequencer is not full-length, perfect chromosome sequences. It's little tiny bits that we then have to make sense of as we go. How we assemble our genomes. So, as I said, these are little bits of DNA, but you want to string them back together to figure out those full-length chromosomes Rasmus is talking about. And that would be really useful. Okay. And then finally, a little bit of work about short read alignments. So how do we take these little sequences and align them to a reference genome as we do, and how we process that a little bit. And as I say, the processing and interpretation will be a little shorter. That's mostly going to fall into Thorfinn's range of this section. So he'll be talking about all kinds of fun downstream stuff you can do once you have a nice alignment that we think is reasonably good. Okay. I'll give you a quick overview of some of the really important things that you need to know about. One is that the genome is incredibly variable. In fact, Rasmus gave you the basic rule for eukaryotic genomes, so humans and flies on this list, but even the bacteria at the top there wouldn't even follow all of the rules as described. And these vary across, so this is showing you the number of base pairs in the sequenced genome. You can see that they vary across orders of magnitude, right? So the bacteria is very small, it's about 4 million base pairs, all the way up to the human at almost 3 orders of magnitude larger. So incredible amount of variation. And of course, some of that is the genes and the things we're really excited about. But actually the vast majority of that is something called transposable elements. And I mention this now because repeats are a really important problem, both in genome assembly and short read alignment. And it's just something we need to be aware of going into this. Is a core feature of biology of genomes that affects the way we interpret those genomes. So the big difference among these genomes is basically things like this, right? It isn't actually the number of genes. Humans have, depending on who you ask, somewhere between 20 and 50,000 genes. It's amazing that we can't decide on a more precise value than that, but we're working on it. But flies have something like 20,000 genes as well. And so the big difference is not actually the proteins. The protein coding content of most genomes is actually quite similar. That is, of eukaryotic genomes is quite similar. So flies to plants to humans all have somewhere in the range of 15,000 to 50,000 genes. Not that different. But the big difference tends to be the amount of repeats. That is the amount of a sequence that's exactly the same. And the big reason for that is what I'm showing you here. These are something called transposable elements. What these are so-called sort of selfish DNA, right? So or RNA in some cases, but they're things that live in your genome and they're parasites. Their goal is to replicate themselves and make more of themselves in the genome. And they do this through a whole bunch of different tricks, which I'll tell you about as we go along in the class. But the important point for now is to know that that exact green sequence or that exact orange sequence can be present many thousands of times over in a genome, almost the exact same sequence. And so if you sequence a chunk of that, it's very hard to know where that came from in the genome, because it could come from any of thousands of different places. Something we need to be aware of when we're analyzing these data is that genomes are not nice, unique sequences, ACTG with different combinations all the way throughout. In fact, it repeats itself many, many times over. And so this can affect both genome assembly because it's confusing to standard assembly methods, but it also affects short read alignment for the reason I just described, which is that, if you sequence a little bit of DNA from one of these, you may not know where it goes. And so that's one of the big challenges right now in just basic bioinformatics, is handling these things competently. Okay, and so before we get into the actual assembly, I'm going to spend some time talking about sequencing technologies and methods. I don't think you need to know the exact details all the way through, but I do think it's really important to understand where the data come from and what the differences among your data types are. So what I'm showing you here is a slide describing Illumina sequencing. And this is the bulk of all genome sequencing going on in the world right now. It's 90-something% of sequence data that's made is Illumina sequence data. So this is the big thing that's running the whole show. There are new technologies we'll talk about that are really appealing for other reasons, but this is the one you need to know about. And so how many of you, so no one's really responding to me, you're not helping, guys. How many of you have heard of polymerase chain reaction, or PCR? Do you know what this is? That's a one, okay. So I'll do the slide slowly. So what's going on here is that this is meant to represent a DNA molecule up here, and it's bound to what we call a flow cell. So a flow cell is an engineered product, humans made this, that has little oligonucleotides, so little sequences ACTG here, that is the exact complement of this side. So they base pair just like your genome base pairs. It has 2 strands that are reverse complements of each other. They base pair the exact same way. And then this thing extends upward from there, right? So you've got DNA from the genome you weren't trying to sequence next to this little chunk of sequence that is exactly the same except the reverse of what's on this flow cell already. So these are called adapters, right? The idea is that we're adapting a chunk of DNA that we want to know about to the technology we have. Okay. So what it'll do then is it'll bend over and do what's called bridge amplification. So you'll go back and forth copying this many times over. That's what's going on over here and here, right? So this primer is copying this chunk of sequence until at the end you have a whole bunch of forwards and a whole bunch of reverses with the exact same sequence. Okay. And then finally, what's going to happen is we'll add little fluorescent nucleotides, right? So each A, C, T, and G is labeled with a different fluorescent marker. So they'd be green, red, blue, etc. And you'd be able to track as these clusters grow, did I add an A? Did I add a C? Did I add a G? Did I add a T? Okay, so at the end of this whole thing, you would have some little chunk of DNA. I didn't say this earlier, but these little sections here tend to be in the range of 300 base pairs. So it's not much, but it's extremely high quality. So we've got a pretty good idea what it looks like, and we got to figure out how to put it back together. Okay. I won't talk as much about this, but I think they're really exciting and really fun because it's completely different. Nanopores are something you may have heard of, and I suspect you've even analyzed some of this data if you've been working on SARS-CoV-2 stuff. Nanopore is really appealing because it's nearly real-time sequencing. And the way this works is rather than building the complementary DNA molecule and asking, did I add an A, C, T, or G? So synthesizing what I'm reading. In this case, we're just threading it through a pore and measuring the amount of, is it just me or anyway? So we're threading this through a pore and measuring the amount of current that's going through this nanopore here as the DNA goes through. And that the amount of current actually tells us what the sequence is because each nucleotide affects this transmission at some rate. And so we have a readout as this thing is threaded through. It goes through really, really quickly. So you'll read, thousands of base pairs in a minute, no question. And the beautiful thing about this is, as opposed to Illumina sequencing, where we go back and forth and have to copy that same thing many times over, with Nanopore, you can just read until you hit the end of the molecule. So you can just keep going and going and going. And the current, I believe, record for the longest Nanopore read is 2.5 million base pairs. So you can read really, really, really long sections of DNA using this kind of technology, which is really appealing for all sorts of assembly things that we'll talk about as we go. Okay. To show you some of the key differences between these data types, what I'm showing you now is a figure from one of my colleagues' recent manuscripts. But basically what you're looking at on the top is, for a given gene, all the different transcript variants. So each of these black boxes would be an exon. This little connection here is an intron. So it's a chunk that is not being transcribed into protein and actually gets spliced out by your splicing machinery, as Rasmus described. And so this particular, we call them isoforms, but version of the gene has this exon, this exon, this exon, this exon, et cetera. But others don't, right? For example, these set at the bottom don't contain this last exon, they just end right here. And of course, if you use short reads and your read maps to say there, you can't tell which of these versions it comes from, right? Because every single version of this gene has this chunk of gene, right? This chunk of exon. But if you had a long read, so for example, a nanopore read, you see them down here, they actually read all the way across, right? There is nothing stopping it. It reads the full length transcript, more or less, although there's some ambiguity at the end here in some cases. And so you get this, what's called a full length isoform. The other big difference, which I think might be a little hard to see, but I'm hoping you can kind of squint at the screen with me, is that you see a lot of little blips here, right? So these nanopore reads have little white dashes in them throughout. The Illumina reads don't. They're a solid black box. What's going on there is the other major difference here, which is that Illumina sequence is nearly perfect. Right? There's almost no errors when we sequence, there are very, very few. And so the alignments can be quite good. And in this case, they sort of match up to the reference almost exactly. Hence, you don't see any of these little white blips where there's a difference. Nanopore, of course, is sort of polluted with those. You can see across these reads that they're quite different from the reference. And so there are many, many little blips here where there's no alignment. Those are the key differences here. And as we go, I'll talk about how we handle those and how assembly can be a little bit different and why very different algorithms have been proposed to handle genome assembly for these sorts of things. Okay. With that, we'll also handle sort of the nuts and bolts of how you get data. So anytime that somebody publishes a sequence where they've sequenced some genome, as a part of being a good scientist, you're required to archive your data somewhere. Where you typically archive your data is what we call in the US a sequence read archive. The Europeans have a version of that. The differences don't matter because they literally mirror each other and have the exact same information. So I'll teach you the SRA version, the sequence read archive version that we have in America, but it doesn't matter. You can get the exact same stuff from each of these. And it's actually a really incredible thing. If you have some time, I would recommend logging into it and try to think of some organism that you think no one would ever have sequenced. And I'll bet that you find that somebody has sequenced it before. There's an incredible amount of stuff on there all the time, and it's just growing at an incredible pace. So one of the big advantages of being a good bioinformatician is that you actually really don't need to make data anymore for a lot of questions. It turns out there's a good chance someone else made that data for you. And I'll show you how to access that data using just standard SRA toolkits, which will be on some Unix cluster that I think we'll set up as we go. And you can sort of download, and I also want to show you at least once how to upload some data, just in case you are someday the bioinformatician working on a project that sequenced a bunch of genomes and you need to be able to put your data somewhere for your publication. Okay, and then we'll also go into some of the more specialized repositories. So I think a lot of you are probably going to be interested in human genetic variation. So there's this really, really large ongoing project called the 1,000 Genomes Project. But basically it's got human samples, population samples, so not just one individual, but typically many individuals from these sites sort of across the globe here. And so the wonderful thing about this is that it will have, this data set tends to have very well processed data. So rather than all the challenges we're going to handle with raw data and how to manipulate and measure that, in this case, you'll often be able to just download something that you can analyze immediately using tools that Thorfinn's going to talk about, and you really can skip a lot of the intermediate steps. So there's a big advantage to working on an organism like humans that's extremely well studied, because we have a ton of information that we've already compiled in very, very nice packages. So we'll show you how to get that, how to manipulate those kinds of data, and just what to do with it. Okay. There are, of course, many others as well. So ModEncode would be the model organism version of this in some sense, not exactly, but I'll go over a few other major repositories that you might want to know about. The goal is at the end, not necessarily that you know how to access each one of these and know where they all are, but you at least know what kinds of phrases to Google to find these sorts of things. So what would I type into Google to figure out where this data lives? And hopefully we can get to that. Okay, cool. We'll talk a little bit about processing raw data. So I think I mentioned this earlier, but the way Illumina sequencing works is you actually shear your genome down, right? So we don't, unfortunately, we can't actually sequence telomere all the way to the other telomere or end to end of a chromosome. It's not possible yet. You can't even extract DNA that long, so you're definitely not going to do it. But what Illumina does is it shears it even more than what we can extract. So it shears down to 300 to 500 base pairs. It then adds these adapters, right? So we go down here, we see, okay, the simple molecular biology is we go through some tricks to add these adapter sequences here and here. And as I said earlier, they've got these little chunks at the end in green and in orange over here. And those match primers or oligonucleotides that we have, that we've engineered onto flow cells. So the reason this is important is that in many cases, if this insert here is too short, so for example, if it was shorter than your read length, if I sequence 200 base pairs and this blue section here is 100, what that means is that my sequencer will just keep going. It's actually going to go into the red stuff and then the orange stuff. And if I'm really unlucky, it'll fall off the end and just, with Illumina data, it'll just tell me there's a G there 100 times over or something like that. So we need to know how to process that and take that out, because this is one way you could add repeats that will really confuse your downstream assembly and data processing techniques. That's one of the main things we'll go into. There's actually a whole bunch of other stuff. So I'll show you some simple and hopefully straightforward quality control methods we use to make these data kind of interpretable and more uniform than they come off the machine in many cases. Okay. Cool. And this can include things like finding sequences that are overrepresented. So for example, if you downloaded a data set from the SRA, you may not know how it was sequenced. It's not always obvious from the submission what they actually did. But you can look at K-mer overrepresentation. That is, do you find the exact same sequence, for example, this red chunk, many, many times across your genes? That would suggest something's wrong here. Or across your reads, it suggests something's wrong. And there's a bunch of other things we'll show you how to find and remove and detect as we go. Okay. And then finally, we'll get into some of the more fun stuff here. So once you have all your reads, you want to interpret them. As I said, they're short, right? It's 300 base pairs at the most, and typically less with Illumina data. You know, that's not much information. We like these to be longer, at least the length of the gene, which would be several thousand base pairs. And so what you want to do is learn how to string them back together. And so the typical assembly process goes something like this. You have a real genome up here. It's shown in gray because it's a myth. It's hypothetical. We don't actually know what it looks like at all at this point, but we might have some idea. But below that, you've got your reads. This is the data we made. And remember, this is something like 100 base pairs, nucleotides of A's, C's, T's, and G's, some sequence like that. And what you want to do is you want to find the chunks that go together. And there's many different algorithms for this. We will work through a few in class just to show you how this works, some of the simpler ones that you can do by hand, or you can at least do subsets by hand. And then I'll show you how to run some of the more fun ones on a Unix cluster to just learn how to make a bigger genome. But basically, the main steps are you take your reads, you figure out how they go together by finding overlapping chunks to make what's called a contig. And then finally, you figure out how those go together. Even if you don't know this little bit of gray sequence in the middle, you might have some reason to know that these two are adjacent in the genome, and you'd call that a scaffold. And I stick as many contigs together as I can. So that's sort of the basic core terminology of genome assembly. We'll go through a few different algorithms for this. The very first were things called overlap layout consensus, which is exactly what it sounds like, but we'll go into the details of how it works a little bit. More recently, the bulk of genomes that we've seen are made by what's called a De Bruijn graph. And that's the kind of thing we can do by hand. It's if you want to look it up, there's some pretty fun basic examples online already, and I'll walk you through a few of those in class. And then of course, there's been extensions of this. So one of the key issues with the De Bruijn graph is that if your reads like Nanopore or the previous version of PacBio reads have an error every 10 base pairs on average, it's very hard to find exact overlapping matches between them. So this sort of problem has been generalized to what's called a fuzzy Bruijn graph, or there's a few other classes of methods that fall under this basic category of fuzzy Bruijn graph, find things that look kind of similar and stick them together. Okay, and then finally we'll get into a little bit of work on I think what has been the biggest impact on genome completeness, which is called scaffolding, which is finding ways to link these contigs that are really, really different and far apart across the genome and realize they all go to one chromosome, for example. And this has been probably the biggest advancement in genome assembly technology the past 5 years, is figuring out how to scaffold these things all together. Okay. We'll also, so finally, once you know how to make a genome, that's the idea behind this, and have an idea how your data looks, you know, if you want to find variation, right? So Rasmus mentioned that what population genetics really is studying the variation amongst individuals. So it's not the genetics of one, it's the genetics of a population or sets of populations. And so what you want to do then, once you have your reference genome, is figure out where the variation is in this genome. And the way that you would do that is to compare two individuals. But it takes a lot of work to make that one really nice reference genome that has chromosome arms that are all assembled and annotated and pretty. And you don't want to do that for every single individual. That's expensive and time consuming. So what you do instead is you sequence a bunch of other individuals with the technology called Illumina again. And then you align those for that first one. You say, okay, but what's different now from that one I saw the first time? Where are all these little differences between them? And so if you saw, for example, this doesn't actually include differences in this example figure. These are all errors. This individual exactly matches the reference. That's not a great choice. But for example, if we saw a column here where this second A here had all T's and all the reads that align to it, you would say the individual I sequenced is actually homozygous for a difference. They have only a T at this site relative to the A in the reference, and we'd call that a SNP, or single nucleotide polymorphism. You've probably all seen that in your reading at this point, I assume. Okay, cool. So I won't go into great detail on most of the algorithms for this. I think the one that's the most fun to explain is called a Burrows-Wheeler transform. It's also the basis of the vast majority of short read alignment that's been done. But it's a really cool way to do alignment and data and genome data compression. So It's also fun because you can do these by hands. That's a really nice in-class example. There are other more sophisticated ways of doing these things or trickier ways, but this is the one we'll go through in class. Okay. There are important trade-offs between alignment algorithms in both sensitivity and speed. So they're not all created equal. Some alignment algorithms are really, really good at putting down exactly the same sequence, but have trouble finding the differences. Others are really good at aligning reads, but take forever. And if you're on, a very small server, you may have some problems running them. And so this has led to some really important problems recently that we'll get into at the end, which is this issue of reference bias. So basically, if the way I find all of my variation in a population is because it's different from a reference genome, I'm much more likely to find things that match the reference genome than I am to find things that don't match the reference genome. And so there's a really important problem here that is imposed by the very basis of how we do the vast majority of the science. So, and then of course, the regions of the genome that have all this variation, all these differences relative to the reference, are probably the most interesting regions of the genome in many cases. And yet, for a long time, until quite recently, we've basically been throwing those out as sort of a standard process in handling these short read data. But now we're doing things like this, and I'll explain what this is as we go, but it's what you'd call a variant graph, right? So rather than a single reference genome that has exactly one version of a chromosome from a population, a variant graph actually knows where the variation is. Right, so in this case, we imagine that you have a population that has 3 differences in this little chunk of sequence, right? It has a C or a G at this site, a G or a C at this site, and finally, this would be what's called an indel. So one of these chromosomes has an A and the other has nothing. It's a deletion. It goes straight from T to C. The idea is, if we put a lot of work into finding all the variation in some population, it should be easy to find that variation again, because I know what it looks like. So I can go back in there and figure out what matches that other version of the chromosome that isn't the reference gene. And this is some really important stuff that's been going on recently. My own lab does a lot of research on this, so I'll probably spend way more time than I should on variant graph stuff. But it's also, I think, really exciting, in particular for finding things like this deletion here, like where the structure of your 2 genomes actually varies. So not just C versus G, but actually they are structurally different. This sort of approach can be really important, and it's done a really good job of finding a lot of this variation. And so it's really exciting for those of us excited about things like balancing selection, which is a process whereby 2 different alleles in a population can be favored, and so they stick around as heterozygotes. What typically happens is lots and lots of variation accumulate in those regions of the genome, and it can be quite hard to study using sort of more traditional methods. Variant graphs are great for that kind of problem in biology, and it's something that we're really excited about doing in the future. So again, I'll probably talk about this more than I should, but it really is going to be the future of short read alignment, I assure you. Okay. This figure is not showing up on my slide. Is anyone seeing this figure? No? It's a blank slide? Okay. So, well, it was going to be showing you how we go through when we find, is there another one? Nope, that's it. Okay. It was going to be the ending slide here. I'm just going to describe what you would do with short read filtration. So normally what you would do is go back in and look at reads that we're not sure about their mapping, right? So I mentioned at the beginning that there are many chunks of the genome that have the exact same sequence across them. And so you can imagine that if you sequence a read that fits exactly in one of those chunks of genome, you can never be sure where in the genome it comes from. And then we typically quantify this with something called a mapping quality. And you can think of that as the probability that that selected site is the correct one for a given read mapping that we report. So that's one of the ways we typically filter short read data, is to say, okay, this site could go anywhere in the genome, it's a problem, and we don't usually analyze those. So this is, anyway, one of the main things we do to clean up these short read alignments and remove a lot of the ambiguity that we get when we do sort of standard data processing approaches. Okay, and it looks like my other figures aren't going to show up either, so that's going to be the end today. And hopefully the next time I'll figure that out in advance. So does anyone have any questions about this stuff? Or I would really like to know if there's other bioinformatic problems that you think you would like to learn about that we could help you with. For example, if you have a project you're going to do or have some idea what you're working on next, I think there's a good chance that I could tailor some of this to help you guys with that.
Well, thank you for your talk. And I have one question. It might be stupid, but I'm going to ask it anyways. Yeah. At the very, very beginning of the talk, you said that we have a problem with genomic sequencing because some of the parts are just repetitive. And there are like many of them, and they are the same. And you called them parasites. And I want to ask a question, whether they are really necessary for the biological body. Do they serve any real purpose? Because if they don't, that, as I always thought, the natural selection will just, might just get rid of them, or not.
Sure. This is a fantastic question. I want to clarify one thing and then I'll actually answer the question. So to clarify, there are many reasons that you'll get repeats in the genome. The most common one is what I described, which is transposable elements, but there are many others versions of this. So for example, often you will get a gene that duplicates. This is one of the main sources of adaptive genes in any organism, is that you first duplicate a gene and then the two copies diverge and perform slightly different functions. That said, the vast majority of repeats are what I described, which is transposable elements, at least within the euchromatic parts of the genome. And so you want to think about selection on these not from the context of the host. So for the host, they're mostly bad. There are some examples that I'll talk about when I introduce that section where they did something adaptive. So for example, if a gene jumps, or if a transposon inserts upstream of a gene, it can affect that gene's expression. So there are cases we know about, probably most famously the peppered moths in England, where a transposon is the reason for an adaptive phenotype. And so there are cases where they're great. The vast majority of the time, they probably do something bad. So right, they might copy themselves and they might copy into the middle of an existing gene that you really need. If it does that, it breaks your gene and suddenly you're a less fit organism. So for the host, it's probably mostly bad. So for me to have transposons jumping around in my genome, I don't want that. But think about the selection from the context of the transposon, right? Its goal, if you will, is to make more of itself. And so it's finding ways to subvert the ways that your genome is trying to control these things. So there's an arms race going on. Basically, your genome often modifies parts of itself to prevent their transposition. And the transposon is evolving to evade those processes so it can keep copying itself over and over again. It's one of the most interesting features of biology, in my opinion, is conflict between different genes. And in this case, you all have to share one genome, but you have very different goals from your transposons with respect to how that genome is going to work and how they'll be propagated.
So do I understand correctly that during this process, the chance of host actually dying is higher, but since we have many occurrences of the gene itself, the chance of gene replication is also higher. So we have two processes which move in different directions. Sorry, my English ain't that good for expressing that. Yeah, and we cannot just let all our hosts die, but still we want us to have as much occurrences of the gene in this host as possible. And we have to actually find the balance between killing our hosts by making them not adaptive and preserving our genome. Of course, I'm talking here from the point of view of genome.
Yeah. I always speak from the host perspective also. So yeah, I don't want these things jumping around either. I think that's fair. But yeah, you want to think of it as a conflict. You don't want the same thing. There's a really famous essay by a guy in England called The Selfish Gene. Have you ever heard of this?
I read it. So that is why the first part of your answer was not that surprising, but
okay, yes.
May I ask you one more question which is connected? Is there a stable evolutionary strategy to not replicate inside the genome, but to preserve one stable occurrence in the genome, but which must be essential for the survival of a host? is it a stable strategy to not to replicate, but to become like the very, very essential part, so that host would not even think to, yeah.
So this is a good question. The answer is there are definitely cases where we call this domestication. So a transposon evolves some function that's relevant to the host and becomes an important part of how the host works. This is also true of some integrated viral copies as well. They basically either evolve somehow or even just come in with a function that becomes adaptive to the host, and they stick around. So that is what you'd call a more stable strategy, but I don't typically think of evolution as stable. I think of it as running on a treadmill. You're always trying to keep on top of this thing.
Okay, so, I get it. So it is stable, relatively stable in some short or middle term.
It's more stable than being a transposon itself, that's for sure.
Okay. Okay, well, I think I got it. Thank you.
Cool. Yeah. I can send you there's a PDF of a really famous book called Genes in Conflict that goes into all sorts of different interesting.
I would really appreciate it.
Yeah, I'll send it to Vladimir or you. Yeah.
Good, good.
All right. Thorfinn?
Okay, so I'll be sharing my screen now. Let's see. Can everybody see this? Okay,
Sure.
and everybody can also hear me, I assume. Yeah, so I'll be running the sessions on the 5th of November, 12th of November, and the 19th of November. And this will be telling what Ross is talking about. And so I have a bachelor in mathematics and computer science and a master in bioinformatics. And my area of research is to develop and extend some of the traditional statistical methods within population genetics, so they work for low coverage sequencing data. And the stuff that I'll be doing in these 3 sessions is to go through this thing like how do we call genotypes. I won't talk about what a genotype is. That is for Rasmus and Ross, I think. And I'll show how to run some of these analyses. So Ross will go through all the file formats, and what I'll go through here today is to show why some of this, we call it NGS data, next generation sequencing data, why this is so difficult to work with from a statistical point of view, why it's difficult to apply in practice. So here I'll just go through how we call genotypes and how we deal with these things in a probabilistic manner. So just as a primer, Ross will go through all this. So I won't steal his thunder, but what we normally do is that we go to the lab and we extract the DNA, and then we sequence it, and we don't get data we can use directly. We get images or small videos. This is converted into FASTQ, and then this is being mapped. And then based on this mapped data, we traditionally call what we call genotype likelihoods, which is the probability of the different possible genotypes. We estimate these allele frequency. I'll go back, I'll define what these are later. We'll calculate frequencies, and then we calculate the proper genotype probabilities. And then based on these, we'll call genotypes. And then based on these, we are using these for actually performing the analysis that we want to do in the downstream, all the fancy stuff that Rasmus talked about. So fundamentally, I would say that we have at least 3 different files. The stuff that is unmapped, which is the FASTQ, and we have the map stuff, which is BAM. We normally use the container format BAM, binary SAM files, which contains all the mapping information. And then we have the VCF file, which solely contains the variable information. And this is, again, this will just be a quick overview so you understand the problems for the later slides. These are the output from the sequencing are these FASTQ files, FASTA files with qualities. So when you have your sequencing machine, it will spit out millions and millions of reads, and each read will span 4 lines in these FASTQ files. There's a read identifier which contains information about which platform and which flow cell and all these things that are not really necessary at this point. It will also give us the basis. That's the sequence of that we have been sequenced. And normally these just contains A, C, G, and T. But in this example, we also have something called N. This is an identifier, which means that the sequencing machine was not able to call a base properly based on these image files. And for each of these bases, there's also an associated quality score. And this quality score gives you a sort of probability or trustworthiness of each of the bases that we have sequenced. Okay, so in the past, when I started working with this, we were using something called the HiSec 2500 back in 2012. This one would generate 600 million reads, and if you wanted to get a human genome, 3.2 billion base pairs and 30x coverage, then that would cost almost 5,000. And the machine itself is also very expensive, 700,000 at the time. They made a new version of this, which was the HiSec 4000 2015, which generates 5,000 million reads, and that half the price for a human genome. And the most recent one that we're using here in Copenhagen, we are running a huge ancient genome lab where we are trying to sequence thousands of ancient genomes. The output from this one is 20 billion reads. And if you wanted to do like a 30x genome, then the price has now dropped to $700 per genome for a 30x genome. So this is, you're getting a lot of data these days, much more than what we used to for a fraction of the cost. But the output from these files, they're huge. I'm in charge of running the bioinformatical part of this sequencing of 1,000 genomes. And for each output from this NovaSeq machine is 6 terabytes of data. And it generates 6 of these per week. So that's almost 50 terabytes of data that needs to be processed and stored and back up. So it's a huge, you need a huge investment if you want to go into this large scale analysis. Okay, so Ross talked a bit about mapping, so I'll not go through this in detail. What the mapping does is that you have your sequence, you do not know where it belongs on a reference genome, so you try to figure out where it belongs on the genome. So the output from this mapping program is the relative position to somewhere on the genome. And as Ross also said, we will also get extra information like where does it match to multiple places? Are there mismatches in your reference? And as Rasmus talked about, the DNA is like a backbone where you have A that matches to C and C matches to G. So you also have a strand information, which one have you sequenced? You also have this information after you've aligned it. And the container format here is this SAM and BAM file. I'll not talk about this. I'm sure that Ross will talk about this for a long time because there's a lot of useful information here. What is important for what I'm going to talk about, which I think is very, very interesting here, is this sort of a quality score, how much we trust the actual, not the alignment, but the basis. This is the quality score. And this is the information at the for the sequencing machine about the probability when it sequences an A, how much does we trust the A? And this is it's Fred scaled. So you take your ASCII value of your quality score, and then you convert this into a proper quality score by taking the ASCII value. And a technical detail is that you need to subtract by 33 because the first 33 characters of your ASCII table is not visible characters. These are the characters that will make your computer go bleep, and it contains spaces and tabs. So you take your, let's say you have a quality score that is encoded as 5. Then you look up an ASCII table and you can see that that has the ASCII value corresponding to a quality score of 20. And then you put it into this equation, and then you'll have a probability of the base being incorrect of 1%. So the probability that the A would actually be correct would be 99%. Okay, so this is the information we will use later on to sort of build the probabilities of the different genotypes. When we have mapped data, that there are a number of summary statistics for sequencing data. The most useful one is the one that is called the depth. That is simply the number of reads that overlap a specific position on the genome. You can also talk about the counts. That would be the number of A, C, Gs, and Ts that you have for a specific position. And we also have the coverage, or sometimes people also call it the depth of coverage. So here the literature is not consistent. I would call coverage the fraction of the genome where we have data, and the depth of coverage being the average depth across all sites with data. So this might not be relevant because a large part of, as Ross also said, there's a large part of the genome that we cannot map to. That it's a repetitive region, so it's a very large, many megabase region where we're just observing the same repeats over and over, and we cannot map to this. So we also talk about, at least when we're using human stuff, we have something called the accessible part of the genome. That's the part where we can actually map to. If we are assuming that these reads are completely random when we sequence them, then we would get something which should follow a Poisson distribution. But this is something we never see when we are sequencing. It always falls off that you have some very few regions where you have extreme high coverage, and then you have a small proportion where you have very low coverage. So I remember I saw a talk some years ago where they said it shouldn't be a Poisson distribution, but a Gumbel distribution. I'm not sure which is most correct. So here we need some definitions, and Rasmus didn't want to say what a gene is, so I won't do it either. Just take your intuition for this. Well, what we need to know what an allele is, and that is like a variant form of a specific gene, right? So that's the overall variance that a specific gene can have. Then the genotype are the alleles within an individual for a specific position, okay? So we are doing all this sequencing, and we have many reads as we see down here. Then the genotype calling would be to infer or determine the true underlying genotype that has generated the data that we observe. This is genotype calling, and this is very
Thorfinn,
yeah.
I'm sorry. Are you supposed to see like your first slide?
Which slide?
So we are seeing like one single slide all the time.
What?
No, it's not correct.
No, I'm on slide 10, I think.
Oh, so now it updated.
It just jumped ahead. It's also not in presenter mode. I'm seeing like the whole.
Okay, just a second here.
I think something's not quite right. Yeah. Thank you, Vladimir. I wasn't sure.
You too. Yeah. I also got some messages on chat. Yeah.
Okay, let's see. I'll try to open it in preview instead.
That may work. Yeah.
Okay, let me kill this one then.
Yeah, you'll switch your screen share to the other one. Yeah.
So is this better?
Yes, now we can see the slides changing.
Yeah. So you didn't see any of these?
No.
Oh, that's a shame.
Sorry, Thorfinn.
Okay, I'll send it out afterwards. Okay, so this is what the distribution looked, the depth distribution for an example we had. So this was where we talked about the genotype calling. Okay, so when you are, there are other, we are working with sequencing data, a platform that was prior and it still exists to within genomic data, something called SNP chip. And this is a very, very useful platform, and this will supply you with the raw genotypes for each position from a predetermined list. So these are, you can use those directly and do all your analysis afterwards. But this is not the case with sequencing data. Can you see the mouse? Good. Okay, let's pinpoint this position. Right here we have the reads. So all the reads that overlap a specific position would be this red one here. Okay. So the fundamental problem with sequencing data is to take the sequencing data we have and then try to guess or infer or estimate or calculate or whatever we want to call it, the truth that the genotype, the original biological genotype that has generated this data we observe. Okay. So for this example here, Ross said that Illumina had a very low probability of error. I think it's because he's used to nanopore data, where for me, an error rate of 1% is very high. I would think that it's around 0.5% in general, but of course it fluctuates along the genome, and it's also dependent on where in the genome we are and which base we are observing. So for this example here, we have sequenced 11 bases. We have 11 reads, and we have 11 bases that overlap this position. We are observing 2 Cs and 9 Ts. Okay. So if we wanted to kind of come up with a good guess for which genotype we had at this position, it would clearly not be an A, right? Because we don't observe any A's. It might be a C because we're observing 2 C's. It might also be something that has to do with a T, but it's very unlikely to be a G. Okay, so how can we? What kind of thinking can we use to try to come up with a better guess of what genotype this is? Well, if we assume that these things are binomial distributed, then we can look at the distribution, assuming that the site is indeed heterozygous. And then we can look at what is the probability that we observe 2 or more, or 2 or less minor bases for this specific position. So the probability here would be taking the sum of the tails that we have, and that would be around 6.5%. But this is assuming that it's heterozygous. This is what we do not know. We want to figure out what the genotype is. On the other hand, if we assumed it was homozygous, the distribution would look like this, and then we would have a probability of 2 or more errors of around 0.015%. So it's not easy to figure out what it is if we want to do this in a stringent manner, because we can use some kind of external information. If you're looking at humans, we would guess to have around the heterozygous site around 1,000 bases. Can we use this information to figure out what the genotype is? So this was some of the questions that plagued the era of next generation sequencing data 10 years ago. And then the solution that they came up with was that instead of trying to guess what the exact genotype is, it's much better to actually calculate the genotype likelihoods. And then the genotype likelihoods is like the summary of all the data you have available, all the bases and all the quality score, and whatever external information you might have, and then calculate a probability of all 10 of the possible genotypes. We have 4 bases, and we cannot differentiate between an AC or a CA genotype. So we have 10 possible genotypes. So what we want to do here is to calculate like the probability distribution of these 10 different genotypes. And then the first one, which was used in GATK or GATK, that came out in 2010, is like the most basic version, which is also the one that is used the most these days, I think. And this is where you are just using your quality scores directly, and then you are calculating the genotype likelihoods. without any sort of error correction or anything fancy. So this is, of course, correct, and it's the proper way to do it if your quality scores are correct. And normally when we have like these kind of introductory courses in sequencing data, this is put out as an exercise where people are asked to do these calculations. So, but I'll just go through this. So in this case, we have a sequenced base that's T with a quality score of 20. Then we want to figure out what the probability of T is, what that is using the quality score. So similar to before with this Phred scaling, we had this Phred scaling, which you didn't see actually. You had this Phred scaling probability of the quality scores. Let's go back so you can see this one. This one. This is the formula for how to obtain probabilities from the quality scores. So you take your raw quality score, you negate it, you divide by 10, and then you take 10 to the power of that value. So here we have a quality score of 20. We divide by 10. So it says 10 to the power of -2. Then we have a probability of error of 1%, right? So the probability that the T is actually correct for this specific example is, of course, 1 minus the error probability. Okay? So what would the probability be that it's not a T? We have 1%, but that needs to be equally distributed among the 3 possible other bases, right? So we take our epsilon, our error rate, and divide by 3. So that would be the haploid genotype likelihood for all the non-Ts. So this is exactly what we use when we want to calculate the diploid version. Then we just plug in these haploid probabilities into this diploid version. Here we are taking half of them, but we don't need to do this because it's a likelihood, so it should just scale to the probability. And this is something that Nikita and at your place have been working a lot on. So also another example we would do if this was a lecture with classes, then they would the students would be given all these quality scores and bases, and then asked to calculate these probabilities for all these values. But so here you can just see it. We have all the different bases. These are the bases that comes from different sequences. They have these quality scores that are ASCII encoded. Then these are being converted into proper quality scores by looking up the ASCII value and subtracting by 33. We put it into this Phred scaling function. So we get these error probabilities. And then we are calculating these values, which is like the haploid version. And then there's this one over here, which means like the other ones, right? Because we are observing T's and C's, but we're not observing G's and A's. Okay, so this would be the genotype likelihood for this example. So this is because if you get more and more data, you get higher and higher coverage, then these probabilities get smaller and smaller. So here in the left side, we are seeing the log likelihood of the genotype likelihoods. On the right side, we are just normalizing them. So they're not on the log scale anymore. And very clearly, also comparing with our intuition, the only genotype likelihood that makes sense are the ones that are comprised of C and T and T and T. The other ones are so close to 0 that we can't even see it when we print it out into this latex sheet. Okay, so this is like the most basic vanilla example of how to calculate these genotype likelihoods, but there are many different ways of calculating these genotype likelihoods. The one that is used the most these days is the one that's implemented in SAM tools. And I'm not sure if that is the best one, but I think it's the most wisely used because it's the one that is used in SAM tools. And this is solely using the quality scores, but it does try to model some kind of it does some kind of error modeling, where it assumes that the first error occurs with a frequency, and then the probability of the error for the next one is higher. There's another version which was used in SOAP-SNP, which is also using the position within the read to model these errors of the data. When you're looking at sequencing data, there's you have something which is called a cycle bias. So you'll have more errors at the ends of the read than in the beginning of the read. This is something that SAM-Tools does not model, and it doesn't model it either in the GATK. Most relevant for what I'm doing when I'm working with ancient DNA is would be a genotype likelihood model that actually tries to model the damage signal that occurs in ancient DNA. This is if you're working with ancient DNA, then it has this deamination where you have an excess of C to T errors in the beginning of the reads. If you're sequencing in one direction, and if you're sequencing in the other direction, it's a G that turns into an A. But this is not something that's, it's only this paper from Vivian Link that's implemented in the software called Atlas, where they are actually modeling this. We have never actually done a proper comparison of this method, and it's something that we should look into. Yeah. Okay. So how?
Thorfinn,
yeah.
Sorry. There is a question in the chat. So Nikita Moshkov asks that SAM-Tools usually used for ancient DNA or not, as GATK is more for usual samples.
Well, I would say that it's people are normally using a GATK if they have very high coverage data and working with human stuff, because their platform is they have a whole workflow defined for how to analyze human data. For non-model organisms, I would say that people are mostly using SAM-Tools. But for ancient data, we are at the current time, we are not really using this deamination signal of this ancient signal. And the reason being that the errors you observe are solely limited to this specific type of variance, which is called the transitions. So if you just remove all the transitions from your data set afterwards, then you will have no ancient damage signal in your data afterwards. Secondly, and they are getting much better in the wet lab. So there's actually enzymatic treatments of your ancient data that allows you to completely purge this ancient signal. In practice, what we do with the ancient data is that we are doing one round of sequencing for specific libraries. And then we're looking at the damage signal, and then we see if there's this damage signal at the beginning of the read. And we use that to sort of authenticate that it is indeed ancient. And then if we need to sequence more, then we do this enzymatic treatment for the same library. And in this new library, where we have already authenticated the ancientness, then we're using that for the deep sequencing. One problem with this Atlas approach for ancient data is that it requires a lot of data in order to recalibrate the covariates that they're using to correct for these quality scores. That's so in practice, I don't think so many people are using it, but it's definitely something that should be looked into. You might argue that the data that's being generated now might not need it because they have this enzymatic treatment, which is called the UDG treatment, user treatment. But there's still a lot of old data going around where it would be relevant to apply. And that is also one of the problems with ancient data, is that there might be, we might not have more of the original material. So it might be impossible to go back and resequence the same sample. Most recently, we were working on a project with, I think it was a Sitting Bull, this Native American, where they were just given like 2 locks of hair to sequence it. And that was all they were given. So all the data that has been possible to generate has been generated. So we cannot go back and sequence more. This is a very different from other sequencing studies with modern data, because there you will always have enough DNA material to do additional sequencing, and you will always have enough data to actually obtain a high coverage genome. Okay. So I'm going into slideshow again here. Can you see this? Okay, so these are the different, the most widely used genotype likelihood approaches or models that is being used. But it's not a fixed method. There's not one genotype likelihood which is superior to the other ones. Okay, so in practice, no matter which tool you're using, if you're using GATK or you're using SAM tools, none of these are doing hard genotype calls directly from the sequencing data. They all calculate this form of genotype likelihoods. So this is the example from before, where we have these 10 genotype likelihoods. These are the only ones that are relevant. So if we were to try to infer or call the true genotype, in this case, we would say that the CT is more likely because it has a higher value, right? This would be like a maximum likelihood estimator. And this is a good estimator, it's fine. But in practice, you might be in the situation where there are two genotype likelihoods which are almost the same. So if you were to use this simple genotype caller, then you would say that the ratio between the two highest one needed to be above a certain threshold. Otherwise, you would say that you wouldn't be able to call the genotype. So this is another effect that you have when you're working with low coverage data, is that you cannot call genotype reliably. Then you will start to introduce missingness into your data. So if you want to only call genotypes with a very high accuracy, then you won't be able to call so many genotypes. Okay, so this is like the most basic maximum likelihood estimator. Another approach would be like in a more Bayesian context. Here we have 10 possible genotypes. So if we were to pick a prior, a useful prior or a good prior might be to assume that all genotypes are equally likely. So the prior in this context would be just 1/10 for each of the 10 genotypes. And if we were to call using this approach, then we would also get that the CT is the most likely genotype for this specific position. And again, you wouldn't just call the genotype and say it's a CT. You would actually look at the posterior probability and only call the genotype if it exceeds a certain probability. A normal cutoff would be to say that you should be able to do it with a 95% posterior probability. So this are two different ways of doing a single sample genotype calling. You can, of course, if you have data from multiple individuals, then you can also start to do more elaborate things. You can start to say that you are calculating a frequency for an overall allele frequency for the specific site, and then use that as a prior instead. That is a different approach. And if we are to use this approach instead, then suddenly we will still call the CT genotype, but then the posterior probability would only be 0.56. In one of the first sequencing studies, I think it was the Panda, when they sequenced the Panda, there they used a different kind of prior where they put a certain penalty on the non-reference allele. So this would be a prior that is very good at introducing reference bias, for instance, like Ross talked about. Okay, so these are actually the state-of-the-art methods for calling genotypes for sequencing data. If we are to compare these, then here we see like a ROC curve where we have a proportion of non-calls against the accuracy. So if we allow to have a lot of missingness, then they will have higher accuracy. But if we want to call a lot of sites, then we'll also have less accuracy. The single sample approach is by far the one that is the worst for low coverage data for 3 to 4x. If you can start to leverage the information from multiple samples, then you get an increased accuracy. That would be to use the allele frequency of your sample as a population prior. Then that is much improved. But then we also see that we have this one up here, which is much better than all other methods. And these are the one that is, we can call that cheating. Because here they are starting not just to use the information within your sample that you have of your population, but they also have population scale data. Here they have reference populations, haplotype. So as soon as you have that, then you have much more prior information, and you can use that as a sort of imputation of all your genotypes. So this is by far the best approach to do if you want to do genotype calling. And this is what is done on all modern human stuff and also ancient human stuff. But this is really dependent on the ability of having a reference data set where you have all this information. So if you're working with studies, if you're analyzing like mammoths or some lizard populations in Africa or something that hasn't been sequenced before, where you don't have any kind of idea about your reference, then you cannot use these fancy haplotype approaches for calling genotypes. So in this sample here, this is what would happen if you wanted to calculate the what is called the site frequency spectrum, and you blindly call genotypes with these two different approaches, and all these are behaving very, very bad. Those never work, but in practice it's not so bad, because people they do the analysis of their data in two separate steps. First, they do SNP calling. So they have an idea about which sites are actually variable, and then you can do more elaborate genotype calling. Okay. Okay, so this is the final slide, and this was just a short introduction to this low coverage data. And this is actually a subset of the slides that will be used on the 5th of November. Where there will also be some exercises. So if any of these things were a bit obscure or unclear, don't worry, that it will be covered in much greater details. Okay?
Good.
Thank you very much, Thorfinn. So any questions? All right, then I guess that's it for today. So let's thank all of our speakers now.
Thank you.
All right, thanks for having us. Looking forward to the next weeks.
Yeah, so we'll meet in a week, and we are looking forward for the next lecture by Rasmus.
Right. See you next week.
Yeah,
Goodbye.
see you. Thank you very much. Bye, everyone.